And our next speaker is Dudley Lamming from the University of Wisconsin, who's, I guess, a rising star in the metabolic field of aging. Some might even say that he's already an established star. So, and I really want to also emphasize that he's a big reason for why we are actually here, because Dudley, together with Will Mayer, started this seminar series, Aging, Science, and Isolation, that is still actually running to some in some modified version. That started during the Corona pandemic, where everybody was sort of stuck inside, and he inspired us a lot in what we're doing now. So, thank you, Dudley, for that. And can you hear me, Dudley?
Yes, I can hear you, and thank you for all those kind words, Morten, and look forward to people joining us next week for our revised version of Aging Science Talks, starting with some mini symposia. Our first one is going to be hosted by Martin Denzel. So I will share my screen. All right, you see that, Morten?
Yes, we see it. Perfect. Go ahead when you're ready. Thank you.
So thank you, everybody. Today I'm going to talk about some of the work that my lab is doing on branched-chain amino acids, and in particular how restricting branched-chain amino acids can promote health and longevity in mice. So when we think about what we can eat, if you're a laboratory mouse, your choices are really simple. You eat the food that's provided to you. But humans are faced with what food author Michael Pollan referred to as the Omnivores Dilemma, the fact that we can choose to eat a wide variety of different types of food. And of course, some of these foods are going to be healthier for us than others. Michael Pollan suggested eating not too much and mostly plants. And of course, that's not the food choices that most people in our society are making. When we think about a diet is really made up of three major macronutrient sources. These are the sources that are used for energy: protein, fat, and carbohydrate. And for many years, decades even, we've been advised that really to optimize our health, what we should be doing is to modify the percentage of different macronutrients in our diet. Several decades ago, the focus was on fat, suggesting that we should eat less of certain types of lipids to protect from cardiovascular disease. More recently, carbohydrates are sort of the big evil in the minds of the public, that you should reduce this because it will improve. If you reduce carbohydrates, you'll be protected from diabetes and obesity. But in all of this, protein has thought to be neutral or positive. And dietary advice overall has suggested that we eat more protein. And this is for two reasons. First, to crowd out calories from fat and carbohydrates. And secondly, because dietary protein is satiating. And so there's an idea that if we eat enough protein, then we'll be less hungry and maybe consume fewer calories. But is this really true? And so there was a really great mouse study published a few years ago by Samantha Solon-Beignet in Steve Simpson's lab in Sydney, where they fed mice varying diets, 25 different diets, varying in macronutrient ratios. And what they found was that the mice that lived the longest were actually the ones that ate a low protein diet. And this was really interesting. And in fact, it actually agrees with a lot of human data. So retrospective cohort analysis conducted on NHANES data suggests that low protein diets are actually associated with decreased rates of cancer as well as mortality in those under the age of 65. Several different studies show that diabetes risk increases as you increase the percentage of your calories that come from dietary protein. And dietary protein has been linked to various other issues, particularly cardiovascular mortality, sometimes just in general, sometimes in linkage with other diseases such as diabetes and obesity. And so overall, this suggested that to us that dietary protein might not be the best thing. And so several years ago, we collaborated with Luigi Fontana's laboratory, at the time he was at Washington University in St. Louis, to look at metabolic outcomes in a human clinical trial, randomized clinical trial of protein restriction. So in this study, we have a control group that had no dietary modifications, their normal diet, and 17% of their calories came from protein. And then a protein restricted group that ate about half that much, where 7 to 9% of their calories came from protein. And these people were fed in the hospital cafeteria, so we know exactly what they ate. And the intervention overall took about 6 weeks. And what we found was really interesting. what we found was that people who were protein restricted had improved metabolic health. So this is only 6 weeks, but they lost about 2.5 kilograms, or a little over a pound a week. And the majority of this weight loss was from fat mass. And so this is not just lean mass loss, it's not just water weight. They're really becoming, having reduced adiposity. We also saw declines in fasting blood glucose levels. Now, whenever we do a dietary intervention in humans or in rodents, we need to figure out whether this is due to calorie restriction. You can imagine that perhaps a protein restricted diet is less palatable, but actually the people here actually ate more. And so I mentioned previously that dietary protein is satiating, and indeed their calorie consumption went up about 10% when they're on the protein restricted diet, yet they lost weight and had improved glycemic control. So I'm not going to go through all of the mouse data that we've published on showing that dietary protein restriction works in mice. Essentially, it does. Mice fed a low protein diet have improved metabolic health overall. They become less fat. They have improved glucose tolerance, as well as a variety of other beneficial health indices. But what we started several years ago and we're continuing to work on is looking at the role of individual amino acids in the response to a low protein diet. And so every protein, of course, is made up of amino acids. We typically focus on the 20 common amino acids in our lab, and in particular, the 9 essential amino acids that are essential for both mice and humans. And today I'm going to focus on some of the most interesting of those, the branched chain amino acid, leucine, isoleucine, and valine. And so what we used was we designed an amino acid specific defined diet where we literally put different amino acids together and focus on these BCAAs. Now, the BCAAs have actually been implicated in metabolic health for many years. It was actually in the late 1960s when it was found that BCAAs are elevated in the blood of people with type 2 diabetes. And more recently, over the past decade, Chris Newgard, Christopher Lynch, Susan Hudson, and others have shown that BCAAs are elevated in obese humans, obese rats, obese mice, that they correlate very well with glycated hemoglobin levels in people, and that BCAAs seem to be very important. In fact, a variety of interventions that promote metabolic health, including protein restriction, actually lower branched chain amino acid levels in the blood. And so our protein restricted humans, we found that their BCAA levels declined by about 10%, whereas other amino acids such as methionine that have been implicated in, say, the health benefits of a vegan diet were completely unchanged. And so something is really interesting about the BCAAs. So our amino acid defined diet setup is as follows. Our control diet has 21% of the calories coming from amino acids. Our low amino acid has 7% of the calories from amino acids, and we keep everything isocaloric by boosting carbohydrates. And then we have our low BCAA diet, where we selectively reduce the level of all 3 BCAAs by 2/3 from the level found in the control diet to the level in the low amino acid diet. Literally, these are individual amino acids. They're mixed together in a big vat and with fat and sugar and vitamins and minerals, and then pelleted by our food vendor Invigo. All the diets are isocaloric, and the calories from fat and the sources of fat are completely constant. And in the case of a low BCAA diet, we can either balance those calories by adding carbohydrates or by proportionally increasing non-essential amino acids, which lets us keep everything isonitrogenous as well. So a major advantage of this approach in doing these things in rodents is we can control very specifically the BCAAs in ways that are very difficult to do in humans. Briefly, some of the data that we've published on these diets show that low amino acid fed mice as well as low BCAA fed mice eat more. So just like the humans and mice on a natural source low protein diet, their calorie consumption goes up when you reduce either amino acids or specifically the BCAAs. They gain less weight. This is young growing C57 black 6 male data right here. And you can see they gain less weight on these diets. And this impacts both lean mass and fat mass, but it preferentially seems to target fat mass. And so overall, mice fed a low amino acid or low BCAA diet are leaner. They also have improved glucose tolerance. So here we fast the mice overnight and then administer glucose at time 0 and track blood glucose levels over the next 2 hours. This is very similar to some tests that are done in humans. You can see if we look at the area under the curve, both a low BCAA and a low AA diet significantly reduce the area under the curve, improving glucose tolerance. So in this work that we published a few years ago, basically what we found was that dietary protein restriction in humans and mice lead to very similar effects on weight and body composition, as well as regulation of glucose homeostasis, and that we could recapitulate almost all of these effects by specifically restricting the branched-chain amino acids, leucine, isoleucine, valine. And so that's work that we published in 2016 in collaboration with Luigi Fontana's lab. Now, since then, we've focused on another question. Since we know that dietary protein restriction can extend lifespan, can we selectively reduce the BCAAs and also improve health and longevity of mice? And so we started this work focused on mouse models of progeria. Progeria simply means disorder of rapid aging. And so we used two different models. Laminae null mice, originally a model of sort of aging. It's now thought to be a better model of muscular dystrophy. They're very small and very short-lived. And we also looked at Laminae G609G mouse, which is a mice that has the same splicing mutation as the majority of human cases of Hutchinson-Gilford progeria syndrome. And also recapitulates the pathology of that disease very well. And so both of these we thought would be great systems to sort of try and rapidly test the effects of a low branched amino acid diet. We used a control diet, again, 21% of amino acids, a low AA diet as before, and a low BCAA diet that's completely isocaloric and isonitrogenous with the control diet. And what you can see here is our laminae null female data. We got a very significant extension of lifespan when we place these mice on a low BCA or low AA diet. You can see we get an almost 90% increase in median lifespan on a low BCA diet. No change in maximum survival overall, but definitely beneficial to the mice. Looking at the G609G model, we found actually a really stunning effect. There was a small effect on median lifespan, but a gigantic increase in maximum lifespan. And so you could see that we get basically about a doubling in terms of maximum lifespan out here, with one mouse surviving substantially longer than even the longest lived control diet fed animal. And so this suggested to us that indeed a low BCA diet had some geroprotective effects. Now, of course, very few people in the world have any progeroid disorders. There's only about 150 people we know of with Hutchinson-Gilford progeria syndrome, for instance. And so we wanted to know about the relevance of this to wild-type animals. And so we started with 16-month-old animals from the NIA aging colony. We fed these mice either a control or low BCAA diet. We looked at both males and females. And we tracked both lifespan and various indices of health span as the animals age. So you could see that control diet fed mice tend to gain weight during this experiment, and low BCA diet fed mice in comparison control their weight much better. They don't gain weight here. This is primarily because we see an increase in fat mass in both males and females, whereas fat mass is controlled in the low BCAA fed animals. This is not due to changes in calorie consumption. Just as in our young animals, our old animals fed a low BCA diet actually eat more, not less, normalized to weight, and on an absolute basis, they're not calorie restricted either. One of the things that, of course, if you're eating more and not gaining as much weight, there's a question about what's going on. They're expending more energy, and so using metabolic chambers, we see a significant increase in energy expenditure in both sexes when we feed animals a low BCAA diet. And this also recapitulates some effects we see in younger animals as well. Both males and females fed a low BCAA diet as they age have improved glucose tolerance, and this is maintained as the animals get older as well. And so overall, we see significant improvements in their ability to regulate blood glucose levels. We used Susan Hallett's clinical frailty index to measure the health span of these mice. So in our laboratory, we scored these animals blinded on 26 different traits. And you can see that both females here on the left and males here on the right, our control-fed animals are increasing with frailty as they age. And that effect is blunted in our low BCAA fed mice. And so the overall effect on these indicators of frailty is a low BCAA diet reduces frailty as the animals age. Now, survival is kind of an interesting question. I'll take a moment to explain this a little bit more in depth. You can see overall there's no effect of a low BCA diet on either male or female lifespan. And so that's very clear in the case of males because these curves basically overlap completely. But something interesting seems to be going on with the females. And so you can see that there does seem to be an increase in median lifespan, but there's also this portion of mice here, almost 25% of the total, that dies very rapidly after the diet switch. And so there's a question in our mind, and we're going to have to do additional work to determine whether these deaths are actually aging related. Both calorie-restricted mice and methionine-restricted mice show very similar effects. Tryptophan-restricted mice have shown some data similar as well, where rodents placed on these diets, when you switch to diets, often have a proportion of the mice die very rapidly as a result of a diet switch and not from aging per se. And so one thing that we found interesting is if you exclude those early deaths where the low BCA mice have a significant early mortality following the diet switch, we actually do see an overall positive effect on lifespan with about 11% increase in median lifespan. And one piece of data that suggests this evidence might be relevant is if we look at all of the mice and we look at cancer, which is the major cause of mortality in black 6 mice, what we see is that while control-fed mice have about 60 to 70% of the animals die with cancer that we observed at necropsy, that is not true for the low BCA-fed mice, where the cancer rate was about halved. And so overall, we still need to do additional pathology and follow-up experiments to look at this, but it seems overall that there may be a positive effect on female lifespan when started at midlife. Now, all sorts of diet interventions work a lot better if you start earlier, and they have greater effects on lifespan. And this calorie restriction does as well. So we wanted to know what would happen if we started a low BCA diet earlier. When we start a low branched amino acid diet earlier, in females, we see basically no effect, but in males, we see a dramatic effect with about a 30% increase in median lifespan in either mice eating a low BCAA diet or mice eating the low AA diet. And so basically, a low BCAA diet completely recapitulates the effect of this diet on male lifespan. Now, females, actually, there's a dearth of literature data in the literature about the effects of protein restriction on female mice. If anything, you could see the low A diet. This is a relatively small cohort, but actually might have a decrease in median lifespan, although that's not statistically significant. So trying to understand what actually mediates these effects, we've done transcriptional analysis on the skeletal muscle of the mice, focusing on this tissue due to the effects of a diet's on fitness. Interestingly, about twice as many genes change in male mice fed a low BCA diet as female mice, and they're very different genes. And here's a principal component analysis. You can see that while control-fed male and females overlap, we pull low BCA fed mice in different directions for the females and the males. When we look at pathway analysis using directional KEGG pathway analysis, what we see is that the pathways that change in males and females are basically completely different. And interestingly, one of our old friends shows up, the mTOR signaling pathway, here in the males. And we actually see at the Western blot level as well that there's a decrease in mTOR complex 1 signaling both in young and aged animals in their skeletal muscle when they're fed a low BCA diet. And this is specific to males. We don't see this in the females. And when we quantify this, you can see it's statistically significant for S6 and S6K1. ULK1, which is another mTORC1 substrate involved in autophagy, has a trend in the same direction. And we don't see any effects on mTOR complex 2 signaling. So to conclude, what we found is a decrease in consumption of branched amino acids improves metabolic health in both young and aged mice. The low BCA diet inhibits mTORC1 signaling in the skeletal muscle of aged males, as well as in liver. And the consumption of a low BCA diet seems to promote fitness and longevity in both progeroid and wild-type mice. And so we think, lots of people take branched amino acid supplements. Obviously, we don't know if it's a good idea or a bad idea. Mice are sedentary. People who are taking BCA supplements are often very athletic. And of course, mice are not humans. And so going forwards, we want to know if humans and mice respond to dietary protein BCAAs in the same way. So I'd like to thank everyone who participated in this work, which was really spearheaded over many years now by Nicole Richardson, a talented graduate student in the lab who just started her postdoc at the University of Washington. I'd like to thank all of our supporters, including the NIA, AFAR, Glenn Foundation, and the Progeria Research Foundation, as well as our great collaborators. Thank you.
Thanks, Dudley. Really fantastic talk. So there's a number of questions online. The first one is from Carl Fleger. Is benefit of low branched-chain amino acid diet reduced as total calories go down?
That's an interesting question, and that's not anything that we have examined in detail right now. So one thing that we are interested in is trying to understand if there's a role for branched-chain amino acids in calorie-restricted diets, and certainly looking at different levels of caloric intake would be interesting. But this is all in the context of ad libitum fed animals.
Okay. There's a question from myself. Great talk, Dudley. Is there an effect whether an amino acid is ketogenic or glucogenic?
Oh, that's a very interesting question. It remains to be determined, but we do think that it's probably more. It's hard to say. So there are changes in blood ketones, and that's something very interesting that we're working on. And I suspect the answer will be yes, but that's something that we're still investigating.
Okay, sounds very exciting. We will keep a lookout of those results in the future, I guess. We have a final question from Regal Houtkoper, which you partially answered, I think, but we can maybe get it elaborated a little bit. Regal writes, great talk, Dudley. Does it matter when you start the diet? Do you think there's an antagonistic pleiotropy type relation?
That could be, so one thing that, we observed is we didn't see as great effect on lifespan, certainly, and it seemed to change in terms of its direction, or rather sexual dimorphism if there was an effect in late life, whereas in early life, it seems to be beneficial. And, there's a lot of things related to growth hormone, IGF-1 signaling, mTOR signaling, where there does seem to be effects that are greater when you start younger in life. One of the advantages, of course, of rapamycin is that you don't see that effect. And so I think at the very least we can say that, even if mTOR signaling plays an important role in these responses, which we believe that it's sort of there, maybe overlaps, but it's certainly different from what rapamycin is doing. And so trying to understand age of onset would be really interesting, I think.
That sounds very exciting. Thank you so much, Dudley. I hope you can go on Slack and then answer some more questions there. There is a huge amount of questions.